Excellent. We have reached the last video and we can think about things that happen in the laboratory. And this will get us a little bit closer to clinical microbiology, which is what a lot of you came here for. So let's think about this. Let's think about growing microorganisms in the laboratory. So here we are going to differentiate between chemically defined media and complex media. These are different mixtures of chemicals that we use as the nutrients for growing microorganisms. And chemically defined media is where we use pure chemicals that we get from a chemical supplier, and we know exactly how much of each chemical goes in, and we use pure water, so we can even control the trace metals if we want to. Complex media are used, they're more convenient for us. We don't have to weigh out as many pure chemicals, but they're different in that the things we put in are complex. They are digested proteins and they are biological materials that are usually industrial byproducts, and they're inexpensive for the most part. We don't know exactly what chemicals are in them, but we know we can get similar batches of them, so we can predict how cells will grow in them. And they're less expensive and a lot easier to use. So here are some examples. A chemically defined medium for E. coli. If you look at this, you see things like glucose. You can buy pure glucose, ammonium phosphate, sodium chloride, magnesium sulfate. These are all things you could look up and know exactly what they are. And that's it. If you put this recipe together, sterilize it, E. coli can grow, which is kind of amazing. Much more commonly, you would grow E. coli in something like this. We have Peptone, which is partially like partially degraded proteins. So there's just a complex mixture of amino acid. You have beef extract, where they have literally boiled beef and taken the water and given us that water. So whatever came out of the beef is in that water, and that would be like nucleotides, vitamins, amino acids, cofactors, trace metals, whatever, and then a few other things. Agar is the solidifying agent that allows us to grow this, to use this on petri dishes. Without the agar, it would be liquid broth, and you could use it either way. You could put it on a petri dish or put it in a test tube, and for the most part, bacteria can grow either way. Another thing to look at is fastidiousness, and I don't know the opposite of this, but fastidious cells have really restricted simple metabolic pathways, and that means they don't make their own metabolic intermediates. They have to find them in the environment. So an example of this would be like our cells with the essential amino acids that we have to get from our diet. We cannot synthesize those amino acids in our cells, even though we need them. E. coli can synthesize every amino acid it needs from glucose and ammonium. All the carbon, oxygen come from there. All the nitrogen comes from ammonium phosphate. Boom. So it can rearrange the carbon in glucose into every carbon-containing molecule it needs. And that is kind of amazing. We cannot do that, right? We have to take those essential amino acids and get them from our diet, because we can't rearrange glucose and ammonia to make that. So we are a little bit fastidious. And we notice fastidiousness when we try to grow bacteria or fungi in the laboratory, because we have to add more different individual pure chemicals when we're using chemically defined media. So here is an example of a chemically defined media for growing a fastidious chemoorganoheterotrophic bacterium, Neisseria gonorrhea, which causes the disease gonorrhea. So you have to give it glucose, but also, let me back up. It needs everything on this list. So you have to give it glucose, but also a complex starch. You have to give it acetic acid, citric acid, and oxaloacetate. Well, these are all things that that are involved in the citric acid cycle. And yeah, the citric acid cycle is where we get lots of other intermediates, like the starting points for making nucleotides and amino acids is the citric acid cycle. And that brings us to amino acids. So Neisseria gonorrhea has its own list of essential amino acids that it has to get from its diet because it can't make these from its own carbon. It also can't make these vitamins or nucleotides. It has to get them from the growth media, wherever it is. It actually needs a reducing agent, sodium thioglycolate. You saw that earlier in the lecture. It reacts with oxygen to lower the oxygen concentration. And then lots of other things, like a source of phosphorus, 2 different sources of phosphorus and potassium. Sodium bicarbonate is a pH buffer. Potassium, sulfur, magnesium, nitrogen, lots of different things, iron. So we might expect to see something like this if it grows inside our cells. And I believe Neisseria gonorrhea sometimes grows inside our cells, where all of these molecules are readily available in the cytoplasm. Whereas something like E. coli never grows inside our cells. It might grow in our gut. In bad situations, it might grow in our bloodstream. It can grow in our urine, but never inside our cells. So it's never going to have access to all of these molecules in its environment. So it has to be able to make everything it needs from glucose, ammonia, potassium phosphate, and magnesium sulfate. So that's the difference between something like E. coli that has really complex metabolic abilities, so it can convert glucose into anything, and something like Neisseria gonorrhea that cannot convert glucose into most of these things. So that talks about the sources of nutrition, but we do more with petri dishes and broth tubes than just grow microorganisms. We can use media to tell us about microorganisms. We can use selective media that's going to allow some bacteria to grow and not others. And we can use like toxins and antibiotics and other stuff and very specific carbon sources that will allow our desired bacterium to grow, but not the wrong ones. And if you go back and look at our list of all the different things that make up a bacterial niche, you can apply all of that to how you grow them in the lab. Yeah, and so we would use this if we have a complex mixture of bacteria. Like if you take a fecal sample, there may be something like 1,000 different species of bacteria in that fecal sample and 1 million different strains or 1 billion different strains of bacteria in that sample. And if you're interested in one of them, how do you find it? Well, you have to go through this process of using selective media that will allow it to grow, but not anything else. We also have differential media, and these will do things like change color if certain bacteria grow on them. So an example would be blood agar, and different bacteria from the Streptococcus genus and others that grow in the pharynx, they will treat the blood. It's usually sheep blood. They'll react with the blood cells differently. Some of them can completely break them down, and what you'll see is a zone around the bacterial colonies where it's completely colorless and they've broken down all the blood cells. Others will break them open and you'll see like green colors from the heme, and others won't break them down, and that tells us a little bit about which ones they are. That helps us identify bacteria in the lab. And you'll see a lot of that in the Labster simulations. And that is it for Lecture 3. Congratulations, you made it all the way to the end. In the next lecture, Lecture 4, we will kind of finish this lifestyle stuff by looking at electron acceptors, and we will look at biofilms and quorum sensing, which are some of the coolest things bacteria do. So that will be next time.